All right, so our next question, actually we have two questions that are relatively similar, so I will combine them. Are vendor qualification packages suitable to address all regulatory questions that could be asked for the process validation? And the second part of that is, are they sufficient to address all potential regulatory questions and challenges?
So I can probably start with that one. I think that the general answer is no, but. So what does that mean? Well, that means that typical vendor packages you're going to find are going to get you through a standard IQ, OQ. PQ is generally left to the end user to describe and how they're going to go about validating that. However, a lot of vendors have started offering this as an add-on, bolt-on service. So I think it really depends on what your strategy is for how you're going to be bringing some of that equipment in. I've seen it work kind of both ways. So it's really kind of an up to you question. But yes, with proper scope definition, you can get vendor packages to stand the regulatory rigor. Most companies choose to do the PQ themselves, though.
Okay, great. The next question is, while there are a lot of benefits for single use, what about the costly activity of leachables and extractables?
I can maybe start if you want, just to say that as a supplier, we provide already extractable or extractable guides. We cannot do leachables because leachable means the compatibility with the drug substance or the drug product, but anything before that is already taken into account during the qualification. So most of the suppliers do have extractable guides. And for instance, as well, BPOG, as the Bioforum group, has defined a protocol, which is now very well accepted in the industry, for the suppliers to test all in the same conditions, so that all the extractable can be in somehow comparable between themselves. So the supplier industry comes with some of the preliminary data, but of course, if you're single use, it depends where your single use is used in the process, because when it's upstream anyway, most of the leachables are anyway filters during your process. So this is also one of the findings we had when studying extractable and leachables.
Yeah, I'd like to add to that as Magali introduced, there are consortia, private companies that have published white papers and standards for establishing extractable materials, and they're being very well accepted. A lot of work went into establishing what solvents are being used and what levels are acceptable. The only thing I like to add is that if you're making a monoclonal antibody or a standard product with standard culture media and buffers, we really believe that these are sufficient, that they've been well researched, and it's what should be done. But if you're doing something a little bit unusual, maybe even in vaccine manufacturing, and you're using a single-use tubing, filter, or container with a different solvent, tonicity, different ions present, or different even hydrocarbon, maybe an alcoholic solvent, then you're really kind of on your own. I don't think that's been examined. So that would be a unique case.
Yeah, we try to have a bracketing approach, and for sure, the end user will have to check that the product in contact is bracketed into the broad range of solvents that have been used for the extractable guides. Agreed.
Yeah, and as an end user, we generally find that this extractable leachables is a lot easier to execute from a cost and a timing perspective than it is to have a full sterilization program like you would if you were running traditional stainless. Just think of all of the utilities and times that you're going to do spending, CIP and SIP studies to basically prove the same thing, that the product contact surface doesn't have any ill effect on the quality of your product. If I can do that in a laboratory setting, where it's easier to control, easier to do, it generally saves me time and money.
One more point, and that is that single-use materials particularly have a different adsorption and absorption or absorption profile for even critical materials in some processes. For example, the majority of single-use containers readily absorb sterols. So if you're adding a steroid hormone or cholesterol as a metabolic intermediate, we know that most plastics will readily take those out of your solution. So it goes the other way too. And some of the white papers, some of the publications describing legibles and extractables address this, and some don't. Another category to consider is particulate matter, levels and sizes and sources of particulates in single-use material. So those three categories beyond extractables and leachables are interesting to consider.